I'm going to do a short video on the Think Pair Share number 2 from DAC number 12, kind of the in-class activity that we did on DNA replication and how to draw the DNA replication fork. So I got a lot of requests and questions about this topic, so I'm just going to briefly show you how I would want you to draw this when I ask for it on the exam. In DAC number 12, I asked you basically to draw me a replication fork, or two of them, because remember, every time you have an origin of replication, it creates two replication forks. So I asked you to include a variety of things. One of them was, the first thing obviously is two anti-parallel DNA strands that are going to act as the template for replication. So what you do for that is you just simply start with your polarity, 5' right, to 3', and then obviously you need your other strand of DNA is reverse and anti-parallel and complement. So that means that this will be 3' to 5'. Okay. So this is the parental strands that we're going to replicate. Now I asked you to indicate where an origin of replication is, and so the origin of replication you can pick anywhere. It's typically in the middle of what we're going to draw. And just remember that the origin of replication, so I'll draw this as replication origin RO, is a sequence of 100 nucleotides, all right, that are rich in AT base pairs, because we want to have less hydrogen bonding so it's easier to pull apart that origin of replication for the initiator proteins that do it. Okay, so let's then expand this a little bit. So as the origin of replication starts, which by the way I'm just going to draw a single line here, the strands will start to separate. Okay, so for example, I'm going to change colors. A DNA polymerase will bind to each strand that starts to separate. Okay, and remember, DNA polymerase only synthesizes DNA in the 5' to 3' direction, which means that it's going to move along the template strand in the 3' to 5' direction. So this DNA polymerase is going to go this way, and this DNA polymerase is going to go this way. And that's what's going to help open up the fork, along with obviously helicase, which is the enzyme needed to unravel the DNA strands. So let's draw a the replication fork open a little bit farther. So 5', okay, and we'll just draw our little kind of bubble here to 3', and then again you're going to have your anti-parallel strand 3' to 5'. So again, the origin of replication was right here. All right, so the replication origin doesn't change. It's always wherever replication began, that's where it is. And our DNA polymerase is started here and started synthesizing in the directions we showed. So those create the newly synthesized strands. I'm going to draw here in green. So 5', all right, to 3'. That's the direction that DNA polymerase will synthesize the new strands. And again, the same thing here, 5' to 3'. And DNA polymerase is still going to be here, all right, making the newly synthesized DNA strand. Now, these are what we call the leading strands. So this is, whoops, it's okay, we can do it in red. This is the leading template strand. All right, meaning that it's acting as a template for the leading strand, and essentially the strand that's made continuously, all right, without breaking up into fragments or without being made backstitching like we're going to show you in a second, is called the leading strand, because this is leading the DNA replication machinery. All right, it's being made continuously. Okay, so the same thing here. This is I'll just write LS. This is the leading strand. Okay, so as DNA polymerase starts opening up these forks, all right, one thing to keep in mind is that each replication has two forks. Okay, here's a fork and here's a fork, right? So this is a replication fork, this is a replication fork, and these forks are moving in opposing directions. So as the forks are opening by the action of the leading strand, what's happening is the leading strand is making a copy here, all right, 5', 3', So what's happening here is as the forks are moving to open up, the leading strand is being made on each side, on each strand of DNA, of template DNA, but this is exposing a region of DNA that's not been copied yet. Okay, here's your region of DNA that hasn't been copied. Now, you can't have a DNA polymerase up here. Let's go back up to this example. Oops, sorry about that. You can't have another DNA polymerase here and synthesize continuously in this direction because it'd be going in the 3' to 5' direction, and DNA polymerase can't do that. All right, so what happens is as the leading strand is being made, you're opening up this region of DNA that can't be copied. And so the way the cell does this is by using the Okazaki fragments. So let me get rid of the little box there. So what happens is as this region of DNA opens up, another DNA polymerase will bind, okay, and it'll start synthesizing DNA 5' to 3', which means it's going to go in this direction, right? 5' to 3'. That's what the polymerase will do. And the same thing will happen on the other strand, is as this region opens up, the DNA polymerase will bind here, and it will synthesize DNA again in the 5' to 3' direction. Okay? So they call this kind of a backstitching mechanism, where as new DNA is exposed, a new DNA polymerase will come in and make small fragments of DNA. And so these are called the Okazaki fragments. All right? So Okazaki fragments, after the scientists who discovered them. And the DNA, fragments. And the DNA that they're being read off of is called the lagging strand, okay? And the Okazaki fragments are also sometimes referred to as the lagging strand. So this is the lagging strand. So the lagging template strand is the DNA that's being used as a template, and the Okazaki fragments can be, I guess you can consider them as lagging strands. And the reason why they're called lagging is because they're not made continuously, they're made discontinuously. They need to wait for a region of the fork to open up on exposed DNA on one side, and then they can come in and backstitch or fill in this DNA in small fragments. And that's why it's called a lagging strand, because it lags behind the leading strand. Okay, so let's go over and draw this again. I labeled kind of a lot of stuff there, so I want to simplify it for you. And we're going to draw this replication fork a little bit later on in replication, so when it's had a little time to replicate. So let's draw kind of a bigger fork here. So here's your replication forks. Remember, there's 2 forks per replication, and they're moving in opposite directions. And if our origin's here, so here's our replication origin, here's our replication origin, all right, you're going to have a leading strand going in each direction. So in this case, the leading strand down here will be going 5' to 3', okay, which means on the complementary strand, it's actually moving along in that 3' to 5' direction, all right? So the newly synthesized strand is 5 to 3, but the template strand that it's reading is actually reading it along 3 to 5. All right, so same thing up here, 5 to 3. And again, you would find DNA polymerase, because it's being made continuously, you'd find DNA polymerase continuously bound to this strand and constantly moving in a continuous manner along the replication fork. And the lagging strands, what you're going to see after a while is a bunch of fragments that are 5' to 3'. So let's just draw several fragments here. There we go. So these are all 5 to 3, right? Because this is the correct orientation. 5 to 3. One thing to keep in mind is always label polarity whenever you draw DNA replication problems, because that way I'll know that you know what the polarity should be. And these are the Okazaki fragments, okay, that are being made discontinuously as the fork opens. Okazaki fragments. So I'll just write Okazaki frag. Now fill them. All right, Okazaki fragments. And same thing here, this would have a series, all right, of fragments. Whoops, 3 and 5 to 3. Okay, so this is essentially what would happen, and as As the fork opens even further, another DNA polymerase will bind here to the newly exposed lagging strand and synthesize another Okazaki fragment. Okay, so this is the basic bubble that I would want you to draw. I want you to understand the issues of polarity when it comes to DNA. I want you to understand that the replication origins are essentially just the point at which replication began, so they will always be in the middle. All right. I want you to understand that the DNA polymerases on the leading strand are continuous, so they're always going to be bound to this strand, and they're going to be moving, all right, they're going to be synthesizing DNA in the 5' to 3' direction, and then there's the Okazaki fragments that are being backstitched behind as the replication fork opens. So this is the think, pair, share question number 2, all right, about the diagram that I want you to draw or be able to draw and understand. All right, and you should be able to label all the parts. For example, if I say where is the leading strand, you should point to this region at least and call this the leading strand. And just remember that this is the leading template strand and this is the leading strand, newly synthesized strand. Okay, so you can call them both leading strands. This is the lagging strand right here. Okay, lagging strand. These are the Okazaki fragments. I'm okay with you calling the Okazaki fragments lagging strands. I just want you to understand that those are the ones that are made after the replication fork is opening up the DNA. These are lagging behind, all right, when it comes to DNA synthesis. So Okazaki fragments are made after the leading strand opens up the fork and exposes a new region of DNA that needs to be copied, all right, in a discontinuous manner. Those are the lagging strands. Where would you find your DNA polymerases? Well, on the leading strand, you'll find your DNA polymerases always kind of bound at the fork region, moving in that direction. And you'd also find a DNA polymerase at the fork for the lagging strand where it's going to fill in the exposed region of DNA. For these Okazaki fragments, we can assume that they're already made, so we wouldn't necessarily find DNA polymerase at these fragments because they've already been made. They just haven't been glued together yet by DNA ligase. So it's a little bit tricky on where to put DNA polymerase on the Okazaki fragments. I'm okay if you put DNA polymerase right here, just to indicate that DNA polymerase would be found there making the Okazaki fragments. But what I'm really going to focus on is you knowing that the DNA polymerase is going to always be bound for the leading strand synthesis there. And this is the basic structure that, or diagram that I really do want you to know how to draw. Okay, so if there's any questions on that, let me know. I'll post another video later on about the last topic that we did on transcription, but hopefully this makes sense and has cleared up some things for you and will help you when you're studying for the exam. Again, as always, email me if you have any questions or come see me.